To survive, bacteria like E. coli have to be ruthlessly efficient with their energy. If there's no lactose sugar in their environment to eat, building a dedicated microscopic factory to digest it would be a fatal waste of resources. In 1961, François Jacques and Jacques figured out how bacteria solve this problem. They mapped out a mechanism showing exactly how organisms turn specific genes on and off based entirely on physical cues from their environment. This diagram maps out the physical components of their model, known as the Lac Operon. First, there's the promoter, which is the start line for transcription. Next to it is the operator, acting as the main switch. Then come the three structural genes, lacZ to break down lactose, lacY to let the sugar into the cell, and lacA to detoxify byproducts. Normally, this entire system is powered down. A separate regulatory gene called lacI constantly produces a repressor protein. This repressor locks directly onto the operator sequence. When the RNA polymerase enzyme tries to move down the DNA to transcribe the structural genes, it hits this physical barricade and stops. Because the path is blocked, the factory stays completely shut down. The bacteria conserves its energy, ensuring those digestion enzymes are never built unless the raw materials arrive at the cell membrane. When lactose finally enters the environment, it seems logical that the sugar itself would remove the blockade. However, the system requires an intermediary conversion step. This animated diagram shows how that happens. The cell always maintains a tiny basal level of the beta-galactosidase enzyme. When lactose enters, this enzyme converts some of the sugar into a structural isomer called allolactose. This specific molecule is the true inducer of the system. The allolactose fits perfectly into a binding site on the repressor. This connection causes an allosteric shift. It forces the protein to warp its three-dimensional shape. With its structure changed, the repressor loses its grip on the DNA and floats away. Now the promoter is clear. The RNA polymerase freely races down the DNA strand, transcribing the lacZ, lacY, and lacA genes into the active enzymes needed to digest the incoming lactose. This chemical trigger ensures the cell only invests resources into digestion machinery when the target nutrient is actively confirmed to be present. There is another variable to consider. Bacteria actually prefer to metabolize glucose because it requires far less energy to break down. If both sugars are available, the cell needs a way to dial down the lactose factory. This is managed by a positive regulation system. When glucose levels drop, the concentration of a signaling molecule called CAMP spikes. This diagram shows CAMP fusing together with another molecule, the catabolic activator protein, or CAP. Together, this newly formed CAP-CAMP complex binds to the DNA right next to the promoter. It acts as a magnetic landing pad, actively pulling RNA polymerase to the start line to maximize transcription. Let's test this logic. Look at this matrix. If both lactose and glucose are present in the environment simultaneously, pause for a moment and predict the transcription level. Will it be off, low, or maximum? The answer is low transcription. The presence of lactose ensures the repressor is removed from the gate, but the high glucose level means CAMP is low. The CAP landing pad never forms, leaving the RNA polymerase struggling to attach efficiently. By combining a negative gatekeeper with a positive volume dial, the bacteria creates a physical logic circuit that strictly optimizes its metabolic output. In molecular biology, the most effective way to prove exactly how a complex machine works is to intentionally break one of its components. Consider the lacIS mutation, which creates a super repressor. This genetic error permanently deforms the inducer binding site on the repressor protein. As a result, inducer molecules bounce completely off the mutant repressor. The roadblock never moves, permanently starving the bacteria. To consolidate this mechanism for your exams, I recommend taking a screenshot of this summary chart. It maps the gatekeeper rule on the left, the volume dial on the right, and the structural gene functions at the bottom. The Lac Operon shows how life functions through these programmable logic gates. These components execute survival code at the microscopic level, automatically balancing energy costs against nutrient rewards.